Last section must have revised your knowledge about different types of phagocytic cells. Now let's look at the production and recruitment of phagocytic cells. Starting with production of phagocytes. As mentioned earlier, professional phagocytes are of myeloid origin. That means they are produced from the stem cells in the bone marrow. Here, hematopoietic stem cells, HSCs, are pluripotent. That means they are capable of giving rise to several different cell types, self-renewing, and immature cells that give rise to all the blood cell types or lineages. HSC migrate during embryonic development to the fetal liver and bone marrow. There they are induced to differentiate further by the large number of growth factors found in these tissues. Included among these growth factors are the colony-stimulating factors, CSFs. Specific CSF induce differentiation of particular cell lineages. In the presence of these various growth factors, including the CSF, the HSC become progenitor cells. Progenitor cells are more mature and less primitive than HSC, and they have some commitment to develop along a particular cell lineage. Progenitor cells cannot replenish stem cells. Under the influence of growth factors, two separate immune system progenitor cells develop: the common lymphoid and common myeloid progenitor cells. Common lymphoid progenitor cells then give rise to lymphocytes that are T and B cells, and also natural killer cells, while common myeloid progenitor cells further give rise to erythrocytes, platelets, and myelomonocytic progenitor cells. These myelomonocytic progenitor cells then differentiate under the influence of CSF into neutrophil and monocytes that further converted into macrophages within tissues and dendritic cells. Now, after its production, let's try to understand their recruitment. The ability of immune cells to mobilize and traffic to where they are needed is central for their functions in promoting immune defense during infection and in driving inflammatory diseases. Here, among phagocytes, monocytes constantly migrate into blood and healthy tissues when they find any infection of inflammation, either directly by surface receptors or cytokines detection. They migrate into that tissue, and within tissue, they get mature into specialized macrophages. Macrophages remain in a resting state unless they are stimulated by signals binding to their receptors. Once stimulated, they start the process of phagocytosis there. Now let's look at recruitment of neutrophils. Although neutrophils make up the majority of phagocytes circulating in the blood, they are absent from normal tissues and will migrate only into inflamed tissue. By the help of interleukin-17 secreted by T helper 17 cells, and by cytokines and chemokines secreted by resident macrophages, neutrophils are recruited to sites of inflammation. Let's look at the closer picture. Cytokines produced by local macrophages stimulate endothelial cells in local capillaries to increase expression of P-selectin and integrins, such as intercellular adhesion molecules, which then attract neutrophils. These modify neutrophil integrin and P-lectin ligands to make the neutrophils more adherent, which allows them to bind to endothelium and undergo diapedicese. That is a passage through intact vessel walls into tissues. During diapedicese, the neutrophil uses its integrin molecules to squeeze between endothelial cells. Then after diapedicese, chemokine-mediated chemotaxis takes place. That is the directional migration of cells along a gradient of chemokines. So the net result of chemokine secretion is the attraction of neutrophils towards the site of infection. To summarize, resident macrophages at the site of infection secrete cytokines and chemokines, which stimulate neutrophil and endothelium expression of selectins and integrins, neutrophil adherence to endothelium in local vessels, and finally, chemotaxis to the site of infection. So this was all about production and recruitment of phagocytes. Now let's have a brief review about receptors present on the surface of phagocytes. Phagocytes use a variety of receptors on their cell membranes during their journey through the tissues and in their encounters with pathogens or damaged cells. There are 4 types of receptors on phagocytes. First, receptors for chemokines and cytokines. They direct phagocytes to the site of inflammation, and once there, prepare them for action. Phagocytes also contain pattern recognition receptors, PRRs, to recognize pathogen-associated molecular patterns, PAMPs, that are present on microbes. Next is Fc receptors. They bind immunoglobulin or antibody-coated pathogens. And last, complement receptors, that bind complement components attached directly to pathogens, to immune complexes or dead cells. So we learnt about production, recruitment, and receptors of phagocytes in this section. Now we'll be moving to the next and most important section that will explain in depth mechanism of phagocytosis. So, continue learning.